Okay, so today we're going to talk about sterile insect technique. The general idea of sterile insect technique is you get like a factory, okay, that takes in some insect, and inside this factory you culture a whole bunch of these insects, and within the factory you have like build a few capabilities. So you build mass rearing of the insect, you build sex separation, so being able to discriminate quickly in a manufacturing sense between males versus females, and then you're able to isolate the males, and then you have a means of sterilization of males, and then 4, you have a way of mass release, like into the wild. Okay, so imagine this factory produces green sterile males, and in the wild you have some population of insects where there's, let's say the blacks will be wild type males and the reds will be wild type females. Okay, so the gist of sterile insect technique is if you can grow enough sterile males and release them into wild populations, you can skew the ratio where there's more sterile males than there are wild type males. And then when the females decide who to mate with, probabilistically, they're more likely to mate with a male that's sterile if they can't tell the difference, okay? So what this does is if, say, again, the females like pick their mates and they end up picking a high proportion of sterile males, over time, what this does, so if this is an axis of number of insects over time, usually this is done week to week. So let's say 1, 2, 3, 4. So periodically, when this is done, you're usually just constantly rearing, I'm talking like millions of insects, millions. And you're releasing millions of sterile males every week. And then what happens is the population will crash because the females will be laying eggs that are not fertile, okay? And then it's like a feedback loop where in the next generations, the subsequent generations, there will be less and less insects. And so the amount that you're releasing becomes more and more and more overwhelming. And so you can actually eradicate populations through this methodology. Does that make sense?
What is this commonly used on with insects?
We'll talk about that. That's kind of like the next thing. But are there any questions about this, the basic premise of it? Okay. So on the test, I usually say, or I have some kind of question that's usually like, what are the 4 things that are required for sterile insect technique? So these are the requirements, okay? So don't forget those. Let's talk about just to answer your question and to go to the next thing, we'll talk about the historical example of this. Okay, so there is a insect called screwworm. Has anybody heard of screwworm? The species name, somebody fact check it. I think it's, somebody fact check this. I think it's Coakley, somebody hominivorax. Look it up quick.
I mean, I can't really say that. It's Cochleo.
Oh wait, what did I spell wrong? Was I close?
It's Cochlymyia.
Wait, what?
Yeah.
Well, whatever. It's something like this.
It's like really long.
Okay, something like this. It's a screwworm. Okay, so I'll talk a little bit about like what screwworms are. So screwworms cause a phenotype or a pathology called myiasis. Okay, myiasis is infestation with flies, with fly larvae. Okay, and there's different forms of myiasis. There's primary myiasis and secondary myiasis. Primary myiasis means that the larvae can go to a fresh animal. In this case, screwworm is a pest of cows. So you must know a lot. You must have heard of screwworm. Yeah.
Yes.
Okay. So
That's the context I know from.
yeah. So screwworm is a pest of cows, and in primary myiasis, they can land. The fly can land on a cow that's healthy, that has no wounds, and they can just immediately start eating the flesh. So primary myiasis dipterans or flies can eat living flesh. Okay, so they will literally like eat the cow alive. In secondary myiasis, it's usually required that there is an open wound, kind of already there, and then that will attract flies that will lay eggs in the open wound, and then they will eat the flesh at the wound site. Okay, but which of these is worse? Yeah, that's much worse. And oftentimes, so the species that can cause primary myiasis are much worse. And oftentimes, if they cause an instance of myiasis, that attracts then secondary flies that make it worse and worse. So it's again, here's a negative feedback loop where it creates a bad pattern that can lead towards death of the cow. Okay, so this used to be a very large problem in North America. And no longer is this a problem because we have sort of solved this through sterile insect technique. Okay, so what they do, this was in, do I have the year? Somewhere I have the year, maybe not. Around the 1950s. Okay, so around the 1950s, there's this guy named Nippling. And Nippling thought of this idea. And he got the idea from some studies that he had read in Drosophila literature, talking about sterilization of males. So his hypothesis was that you could use the sterile insect technique to get rid of screwworm in North America. Okay, so what he did, and this is in collaboration with USDA and the US Army, what they did is, this is in Texas. In Texas, they essentially like set up one of these factories, and in the basement, they had a chamber full of radioactive cobalt, okay? And they would take like a canister that was filled with, they would grow the screwworms, they would put the pupae in this canister, and then they would drop the pupae through the radioactive source, and as it passed through the radioactive source, it received a dose of irradiation that sterilized the pupae, okay? And then they would collect the pupae, and then they would put them in an airplane. Okay, they put them in an airplane, and then they would fly, and they would literally drop these in the millions out of the airplane in areas where there were screwworms. Okay, so the first place that they tested this, so this is the scheme. Again, you have to have a means of mass rearing, you have to have a means of sterilization, you have to have a means of, in this case, they didn't actually have to separate males and females, but then they had a means of loading them and mass releasing them, okay? So they have the things they need. And if I was to draw a map now of America, here's, let's say that's Florida, Texas, Central America, South America, something like that, okay? So there is an island right around here, just north of South America, called Curacao. Okay, and so they first got permission to test this hypothesis or test this theory of whether this would work or not on this like remote island in Curacao. This is just worth pointing out because if you're ever in the future gonna test some weird like biological scenario, it's a good idea to test it on islands. Why?
Because you're not going to devastate like the whole population. Or
Yes,
just isolates.
exactly. That's the premise of the Jurassic Park movie is just keep everything like on the island. If you're going to test something weird, it's actually a really good idea. Test things on islands. Okay. So they first tested this on island, in an island where they would grow cattle. and they were able to fully eradicate all screwworm on the island in Curacao as a proof of principle, okay? So then once they eradicated all the screwworm there, then they decided to start to eradicate in United States, okay? So where would you start in United States?
Hawaii.
Hawaii, okay. Well, that would actually be a really good idea. Do they have cows in Hawaii?
One of the largest Angus ranch in the U.S.
Is in?
Mount, or on like the Big Island.
Interesting. Do they have screwworm there? I bet
I
they
would
don't.
assume I've never seen it. I think it's completely gone.
Yeah, I bet they don't have it there. So it's probably not there. But if you're on mainland, where would you start?
I would say Texas.
That'd be harder because it's a vast, that's a very vast territory.
Let's go to Florida.
Yeah, so that's where they started. They started in the tip of Florida, and the premise was that if they could eradicate in the tip of Florida, then they could slowly push the screwworm infestation back. So again, somewhere in the late 1950s, they're now have permission and they're releasing these screwworms, and it's working, okay? And they're eradicating the screwworm, and they keep pushing it back, and then eventually they did push it all through Texas, through Mexico, so they were releasing in Mexico, all the way through Central America, and then right around here, there is Panama, okay? And here is essentially where they stopped, because they can maintain, I don't know what the hypothesis was, why maybe they didn't have permission to eradicate all the way through South America, plus that would probably be very, very difficult. But currently, screwworm is now eradicated throughout North America, all the way down to Panama, and we maintain these USDA facilities. So there are still factories in Panama that are rearing these screwworms and continuously releasing these every year to hold back sort of the infestation that might come up out of South America. So this was the first, usage case of sterile insect technique. And this is kind of the most famous thing that entomologists have ever done. So if you're an entomologist, we're really proud of this, because this is one of our real big significant contributions. Okay, this was also, the eradication of screwworm has also been replicated multiple times in multiple places. So later there was a case, an outbreak in Libya in the 1980s. And when there was an outbreak, because we had known that we could, or when I say we humans, had known that we could use this tactic to get rid of screwworm, they are able to implement sterile insect technique and get rid of screwworm in the outbreak in Libya. So this has been utilized multiple times and still is constantly being done from the USDA, and it works really well, especially for screwworm. So that's the first usage case of it.
So are they only native to the Americas? Or how are they, where do they originate from?
The screwworms?
Yeah.
Oh, you'd have to do a life history search on screwworms for that.
Yeah. Have they
Yeah.
just kind of spread out all over the place now?
Yeah, I don't know.
Do you think maybe they avoided South America because they didn't want to introduce a big insect population to the rainforest?
I bet it was just not practical. They probably just, again, South America is a vast continent. So it's probably just okay, we got rid of it in North America, we did our job. It was probably just really cost prohibitive.
Do they still use the cobalt now?
Yes. So they still use cobalt for screwworm, but that's what we'll talk about next. So the next usage case of this, what would you try to maybe use this on?
Mosquitoes.
Yeah, that was the next hypothesis, was to try to use this for mosquitoes. And this is kind of an ongoing quest. So the difference between mosquitoes and screwworm is that, which is sort of elaborative of an important point about sterile insect technique, is when you irradiate screwworm, screwworm still can maintain its fitness even though it's sterile. Okay, but mosquitoes are a little bit more sensitive to radioactivity. So when they originally were trying to do these experiments with mosquitoes, they would produce these sterile males. Let's say again, the greens are the sterile males, and the blacks are the wild males, and the reds are the females, okay? When they irradiated mosquitoes with the radioactive sources, the females were able to tell. So many insects actually have elaborate mating rituals and mating biology that allows them to assess the health and the fitness of the male. And so when you do this with mosquitoes, if the males were kind of like sickly because they're irradiated, the females were able to tell, so this is called mate discrimination. And the females were able to actually like selectively decide and find out, okay, which were the healthiest males, okay? And so that, what do you say? That detracts from the ability of sterile insect to work if there is a significant mate discrimination in the species, okay? So it didn't quite work the way that people wanted with mosquitoes right away. So they had to develop other technologies. So,
Could you sterilize like both of them, the females as well?
could you sterilize both of them?
So they just like wouldn't mate, or?
So that's an interesting question. If you sterilize the females, you're no longer able to produce them anymore.
Well, that's what I'm saying. Like, are we trying to get rid of them? So it's like, or are we just trying to?
You're trying to rear a whole bunch of them in a factory, sterilize the males, release them so that you can eradicate in a local area. I don't know if I answered your question.
Well, I'm just confused. It's like if you sterilize the females, then why you wouldn't even have to worry about like doing it slowly? Wouldn't it just be like a rapid sort of?
Sterilize the females in the field?
Yeah.
Or, okay, so how are you gonna sterilize females in the field?
No, I meant like not in the field. Sorry, like put both males and females in those factories and then release them.
You're saying release both males and females that have been sterilized. But then these females are still gonna be looking for mates and they're still gonna reproduce. So there's gonna be huge selective pressure that favors these females and then these will just die.
Yeah.
So that won't crash the population. Does that make sense?
Yeah.
Okay.
I get it now.
Okay, I was able to do that. Okay, let's see. So for mosquitoes, because the cobalt was not working very well, they had to figure out different ways to sterilize. So it's so sterilization mechanisms. So there are other ways to sterilize. So one of these is the radioactivity. Other mechanisms of sterilization, I just wrote all these out. Where did I write them out? X-rays. X-rays are a little bit less damaging, but can also do sterilization. You can do genetics. So there are genetic ways to sterilize insects. At 4 is Wolbachia. We haven't yet introduced Wolbachia. It'll probably be coming up in the next subsequent lectures. Wolbachia is an endosymbiotic bacteria. So it's a bacteria that lives inside of insects. And it has a unique phenotype that it can sterilize the insects in a phenotype called cytoplasmic incompatibility, or CI. Okay? We'll have a whole lecture on Wolbachia later. So these are some other ways that you can sterilize insects with. And so for mosquitoes, they kind of had to figure out and test other ways. So right now, they are using Wolbachia, and they are using genetics, and some people are using X-rays, okay? So all these have variable success, and everybody's kind of like fighting to figure out like what's the best way to do it. It's probably actually also just worth talking about the process in mosquitoes. So for mosquitoes, when you have the factory, let's see, what are the things, means of mass rearing? So mosquitoes are pretty easy to rear. You just have tubs of water, and you hatch the eggs in the water into larvae, okay? And then when the larvae go to a pupal stage, so the larvae kind of look like this, and then the pupae kind of look like this. And the nice thing about mosquitoes is the pupae have sexual dimorphism at the pupal stage. So the females pupae look like this. They're fatter, they actually have eyes. The female pupae are fatter, and the male pupae are smaller. So they utilize this sexual dimorphism to separate out the males from the females. So what they actually do is they take 2 glass sheets. So imagine this is one thin glass sheet, and then you have another thin glass sheet, okay? And between these glass sheets is some angle, theta, okay? And if you set the theta at the right angle, and there's an opening here on the bottom, if you set the angle of the glass plates at the right angle, you can just take the tubs that are holding the larvae, and then you dump them through the glass plates, and the fat females get stuck in between the plates, right where they're wedged together, and the bottom is perfectly set up so that the skinny males pass through, okay? And so if you do this iteratively, so you do this one round, and then you take the tub, and you do it again, and you take the tub, and you do it again. If you do this maybe like 3-4 times, you can get like 99.9% accuracy of males versus females just through simple mechanical separation. So you can see how this is very easy to scale up and manufacture in a facility. Okay, so that's how they do the sex separation in mosquitoes. Okay, now we can actually talk about, let's see. I'm debating like, how do we go into the genetic mechanisms now? And then we can talk about the release. And then when after they're sterilized, so in the case where they're infected with Wolbachia, you don't have to do anything because Wolbachia just sits in the organism. So you just collect them, and then what they do is they put them in a van. And the van has these little like ejection ports. The companies that do this are Verily, which is owned by Google, and Verily is releasing in about half of the United States, 24 states. They have EPA approval. This is big in Florida and California in particular. And it's also big in China. They're doing this. So this is like a big thing. It's worldwide now. It's almost everywhere where they're doing these like sterile insect technique releases to crash local populations in cities. They did a sterile insect technique in Rio, Brazil, before the Olympics. So we'll talk about that one next. Okay, so we'll talk about the genetic mechanisms now. So, okay, there's a few of these. We'll talk about two. Today, we will talk about Riddle and precision guided sterile insect technique. These are two of kind of like the newer, this one's a little bit older, this one's a little bit newer. Okay, so Riddle, so now we're getting into the genetics. Riddle stands for release of insects with a dominant lethal. Okay, so let's think this through. What does dominant mean in genetics?
It's the phenotype that like takes over.
Yes.
It's the
In relation to a copy number in some way. Tell me about the copy number.
Higher copy number.
So if you have, if it requires 2 copies in it, so this is where mosquitoes are diploid. So the context of the genetics is we're talking about a diploid insect. If you have 2 copies of an allele to get a phenotype, is that dominant or is that recessive?
Do you need 2?
Yeah, that's recessive. So dominant then is if you have 1 copy, that 1 copy is sufficient to give you the phenotype. So everybody remembers dominant versus recessive, right? That makes sense? Okay. So we know what dominant means. So then what is a dominant lethal? So let's say we have some gene, which is a DL, dominant lethal. What does that mean?
It kills the organism with just 1.
Yes. So it kills with 1 copy. Okay. So now we understand what a dominant lethal is. Okay. So then the next sort of interesting question is, if you were asked by your principal investigator to create an insertion on a mosquito of a dominant lethal that kills with 1 copy, how would you create that transgenic insertion, and what would be the problem that you would encounter?
How are you not killing the bug itself?
Yeah, how can you possibly insert this? If you successfully insert this, won't the organism just die? And then you will not be able to recover and rear your organism. So what do you need to do to the construct to make this insertable?
You have to turn it off until it's in the presence of something?
Yes. Okay, so good. So the insert has what's called the TET promoter. So the TET system is just like all the other sort of genetic circuits we've talked about, where you add something, it turns it on. you remove something, it shuts it off. So it's very similar, but there's two different types. There is a tet on system and there's a tet off system, okay? In the tet on system, you add tetracycline and that turns the promoter on, okay? In the tet off system, you add tetracycline and that turns the gene off. So just understand, this is actually a very, very, very common not operon, but promoter system. You'll see this a lot in synthetic biology, people using this TET system because it works pretty well. And tetracycline is pretty easy to get your hands on because it's an antibiotic. But also don't get confused. They're not using tetracycline because antibiotic properties. They're using tetracycline because it's a ligand that regulates this promoter very well, okay? So in the Riddle system, they're using the, hopefully I get this right, they're using the TET off system. So that means if you want to make the transgenic insertion in mosquitoes, what do you need to do? In the mosquito genome. So again, we've developed a promoter under which we can regulate. So how do we develop the insertion and select for, it's an interesting question about selection. What do I want to say? The mosquito life cycle is eggs, larvae, pupae, adult. These three stages, egg is kind of like, maybe not, but these three stages are aquatic. That means they live in water, okay? So if you're gonna make this transgenic insert and you have to culture these things as they hatch in water, all they have to do is add tetracycline to the water. And if there's tetracycline in the water, then the gene is off in the TET off system. So it's suppressing the expression of the dominant lethal. Does that make sense now? How you can get a transgenic insertion of a dominant lethal that won't kill the insect in a way that you can culture the insect and rear a bunch, okay?
So when tetracycline is present, the gene is off.
That's correct.
Okay.
In the TET off system, not in the TET on
Okay.
system.
Okay.
There's two separate systems. Okay, so we understand that. Okay, so now imagine what you do with this. So let's discuss the, I'm gonna leave those up, the genetics more. So we make a mosquito, okay? Let's say this is a male, and its genotype is Riddle. Are we gonna want a heterozygous or a homozygous if we're gonna be doing releases into the wild to get some kind of a phenotype? Yeah, we want homozygous because if it wasn't homozygous, you cannot guarantee, unless it's homozygous, you don't guarantee the inheritance of that dominant lethal, okay? So the genotype of the Riddle mosquitoes that they rear in the factory, okay, looks like this. It's homozygous Riddle, okay? And the mosquitoes in the wild, in the field, are essentially wild type over wild type. Let's say this is a female. Okay. So if you rear a bunch of these males and you release a bunch and you get this cross, okay, that means that the F1s are going to be, in every case, they're going to inherit at least one allele of the Riddle. So it's going to be Riddle over wild type. That's going to be their genotype of all offspring that are mated or created by mating of this Riddle homozygous mosquito. Okay, these are going to be eggs that come out of this female, and they're going to be laid in water in the field, which is going to be some pond or some puddle or some tire or some hole or some sewer, etc. And so what's going to happen to the dominant lethal then in that setting?
There's not tetracycline present.
Yes, there's no tetracycline. So now the gene will turn on, okay? And as soon as the gene turns on, then the killing gene activates, and so all the offspring then just die. Okay? That's riddle.
So how far through egg formation do they even get? Do they gastrulate?
So that's actually like a point of engineering. In this case, it's the dominant lethal. If I remember correctly, the dominant lethal actually kills them at the late larval stage. And they say that they specifically engineered this because it's beneficial that the larvae survive, because then they're competing, creating competition against the wild ones, making it harder for the wild ones to survive. So you get sort of like an added effect. And then as soon as they're like about to pupate, then the dominant lethal kicks in and turns on. And the way I think you have to fact check this, but I think that the way that they do that is, in addition to the TET promoter, I think they have a larval promoter.
Like a Dyson?
I don't know what specifically it is. You'd have to look at it. But I think they have actually like technically kind of a double promoter system that makes it a stage specific killing. But all you'll be required to know for the test is the concept that there's this TET promoter, and when they're in the wild, the dominant lethal will have the ability to turn on because there's no tetracycline in the water. Okay, so that's riddle. Let's see. We can talk about the next system now. So the next system is precision guided sterile insect technique, PGSIT. Okay. PGSIT utilizes Cas9, CRISPR-Cas9. Okay. So imagine you create a mosquito that its genotype is homozygous Cas9. So it's got some Cas9 insertion, probably some constituent, I don't know the precise promoter, but it's probably a constitutive promoter. For now, just know Cas9. Okay. And then imagine you create, so this is transgenic mosquito 1, transgenic mosquito 2. So in this case, in precision guided sterile insect technique, you actually need to build 2 transgenic lines. So you need a Cas9 line. And then in the second line, okay, they actually have 2 transgenic inserts. So the genotype, I'll use green, do I have blue? Orange. Okay. We'll do guide RNA 1. So it's homozygous guide RNA 1. And somewhere else in the genome, it's homozygous guide RNA 2. Okay. And one of these causes, one of these guides, let's say green, targets a female specific essential gene. So some gene that's required for life if you're a female. And this other guide targets, let's say orange guide targets a gene that would be required for sperm fertility. So some sperm fertility gene, where if you get a knockout of that gene, you're sterile. You don't produce sperm. So there's this weird second line that has double guides. Okay. Now when you do the mating, so this is all in the factory, in theory. This has not been field tested yet, I don't think. So in the factory, they then mate these 2, okay? That produces the next genotype. In this case, what they're actually collecting are eggs. So they'll collect a batch of eggs whose genotype is now Cas9 over some wild type guide RNA orange over some wild type and guide RNA green over some wild type. So now these eggs are like triple transgenic. They have triple transgenic inserts because of this mating, okay? And so now what's gonna happen, tell me phenotypically what will happen as these RNPs assemble. So you're gonna get two RNPs that assemble in these eggs. You're gonna get the green and the orange. So much color today. It's a rainbow. Okay, so you get these RNPs in the egg. Now tell me what phenotype will come out. What will happen if you do this?
Aren't they gonna cut like the orange and the green because those are essential genes?
So what will be the phenotype? Yes, you're right. Like these are going to be active. They're going to go to their targets. They're going to execute the cutting on those target genes. You're probably most likely going to get a knockout of those genes. It's going to disrupt the gene function. So what will be the phenotype of these eggs?
A completely infertile population.
They'll be infertile. That's one phenotype.
And there'll be no females.
What?
No females?
Yeah, there'll be no females. It'll be female lethality. So every single one of these eggs.
Wait, so this kind of is like what I was asking about earlier, where with females, if you could just
Kill the females.
yeah.
That's what they're doing.
Yeah.
So in this case, let's say you had this one was a female, this one was a female. It's just going to kill all the female eggs. They're just going to die. And all the male eggs will be, which one is the orange? They will be sterilized, okay? So then what they do is they collect all these eggs, and then they take these eggs and put them in packets, and then you can distribute the eggs out into the wild. So for mosquitoes, you can, what they hypothesize, you could take these batches of eggs, attach them to a drone, and then a drone could fly out and deposit these eggs in different ponds and different pools. And then essentially the only thing that's gonna hatch out are sterile males, which will then initiate the cycle of sterile insect technique. And so you can just keep producing eggs and keep releasing eggs in mass. That's precision guided sterile insect technique.
I think I just didn't word my first question correctly. I think that's what it was.
Yeah, that's okay. Okay, are there any questions about that? How much time do we have left?
Why do you think like drones? Is that just
That's a good question. So, depending on the insect, so the thing about mosquitoes is mosquitoes can breed in very tiny like ponds, tiny little pools of water. So, is it easy or hard to it's very difficult. What I want to say is it's very difficult to accurately distribute the sterile males in a way where you can get good suppression of the population if they're in like these weird patchy spots. So the idea with the drones is like if you're in Africa and you're like in the bush, you could fly these drones to find all the local water sources real quickly and then release in the local water sources.
they can go
Yeah,
far?
and it could be automated. It could be like roboticized. That's one way. Or I mean, maybe you don't have to use the drone. Maybe you just take the eggs and you just like release them out wherever you think.
Has one so far been more like effective over the other? Or
So this one's much newer, and this one has. I have to be careful what I say because I don't want to offend anybody in this field because I actively work in this field. What I'll say is that I think this is going to be much harder
That's
to
what I was
get
thinking.
approved
Yeah.
because you're releasing active Cas9. That's going to make people very nervous. If
Yeah.
you're putting Cas9 in mosquitoes and you're releasing that, plus you're releasing like different guides. Every single guide you add, remember, adds a little bit of off-target effects. You don't necessarily know what's going to happen. Okay, and if you're releasing millions and millions and millions of insects, what do you think the percentage of accuracy like is this of the killing and the infertility? Is it 100%? Yeah, it's probably not 100%. Maybe it's 99.99. But that's not good enough if you're releasing millions and millions and millions because there's going to be massive selective pressure for the insect to survive. And so a single mutation could evolve resistance against this. So for example, what would be a mutation? That's actually a good question to talk about. What would be a quick mutation that would instantly evolve resistance against this? You could mutate the DNA in a single spot that would give you instant complete resistance. What would you have to mutate? So let's say,
Like this is the RUP, they can't cut, right?
let's say here's the sperm fertility gene, okay? And let's say the guide, the orange guide, targets right here. And let's say, so this guide, this RNP, is gonna every time try to come to this exact spot to cut the DNA. If you get a single SNP mismatch, mutation right there in that gene, that guide will no longer recognize that site. So you have now completely lost all infertility. And now this is now an insect that is reproducing just as normal as any other insect, but now it's carrying and spreading Cas9 alleles and guide RNA
I was
alleles.
thinking, I was thinking like you'd have to mutate like the guide RNA or like the Cas9, so like there isn't like any production of it anymore. But I guess that'd be harder than just like making a SNP.
Oh, I see what you're saying.
Yeah. Because I feel like
Yeah, if
it's
the
kind of
Cas9, like if the
foreign.
Cas9 mutated, like that would disrupt the ability.
Well, also since it's like a foreign body, like wouldn't the fly like?
But my point is that this is the easiest thing to immediately mutate to get complete resistance, because you just need a single SNP. And a single SNP, it could even be a non, there's silent mutations that don't change the amino acid but do change the sequence. It could be a silent SNP that changes nothing about the fitness, except it completely makes it fertile now all of a sudden. So I think this is going to be really difficult, but it's kind of like, I don't want to say it's just like a nice idea, but I don't know. We'll see. Like, well, I'd like, it'd be interesting. Hopefully it'll work. I don't know.
What?
Riddle, for context, Riddle has been tested and Riddle does work. They used it in Rio to suppress for the Olympics, but what they did discover. So in the case of Riddle, how much time do we have left?
It's
5 minutes. Okay, so we have enough. So we can like talk about problems. Like all these things have pros and cons.
Yeah.
So far, the most well utilized, I think, and the most adopted is probably the Wolbachia. So we'll have a whole separate lecture on that. That's what I study. So I have a little bit of bias there. In terms of the Riddle works, and it works pretty well, but it does have some problems that people are starting to identify, I think, that they might be addressing. But one of the problems is what's called introgression. So just like dogs have different strains and different breeds, insects are the same way. So you might have, imagine like mosquito 1, mosquito 2, let's say this is mosquito 1, mosquito 2, and then let's say this is the Brazilian mosquito, Brazilian mosquito 3, okay. So these are 3 different strains, 3 different populations of mosquitoes. And these different strains of mosquitoes, just like dogs are different, these might have different abilities to transmit diseases. That's called vector competence. So some of these might be very, very good at transmitting disease, some of these might be bad. So let's say you had strain 1 that was good at transmitting disease, and you had strain 3 which was bad. And for whatever reason, like it still transmitted enough disease that you're trying to get rid of the Brazilian strain, okay? So originally when they did this, they were making the Riddle inserts in sort of like laboratory strains of mosquitoes. And I'm not saying that those laboratory mosquitoes were like particularly good at spreading disease. I'm not saying that, okay? But I'm saying that they had a certain breed of mosquito that they're making these transgenic inserts in that's different from the wild strain. And when they did the Riddle releases, so when they did these matings, let's get rid of the strain 2. When they were releasing these insects, let's say this one is red and that one is orange. They would go, after they did the releases, they'd go collect field samples of mosquitoes that were still there, that were still surviving. And they would find that some of the genes from the Riddle strain had introgressed into the wild population of the mosquitoes. So essentially some genes, some SNPs, some genes, alleles that make this strain different, were now all of a sudden appearing in the wild type strain. So maybe you have a few genes, whatever, from this strain. What does that suggest?
You could actually create mosquitoes who are more apt to spreading disease?
Yeah, that suggests, yeah, okay, so wait, before we get to that, the first thing that this suggests is that the riddle killing phenotype is not 100%.
Yeah.
Some of them are surviving, and some of the survivors are mating into the population and exchanging their genetic information into the population. And so now your point that you brought up is exactly right, that now all of a sudden you're changing that population in ways that you can't comprehend. You're changing the genetic structure, creating a different breed of mosquito. And it's entirely possible that that different breed of mosquito might be actually be like worse than the original mosquito that you had. Like you're breeding mosquitoes.
Is it similar to like BT where it, like riddle might be highly specific to like whatever strain?
What do you mean?
Like you know how BT is like really specific to like how, like what plant or like whatever it like operates with? I was wondering.
Oh, like could this be, like there could be it's possible that there could be strains that would be more sort of like resistant against dying from the riddle mechanism.
Yeah,
That's entirely possible. Let me just sort of like also correct the record. This also just has an easy fix. What's the easy fix to prevent this that they're now doing? So I don't want to communicate that they're still doing this, they're not doing this. What's the easy fix? So instead of creating a riddle strain in the lab from a weird breed, what would you do instead?
You take from the population.
You take, so now the better way to do this is, and this is what they do now, so it's a little bit, they're improving. What you do now is you go to the wild population that you're gonna be releasing into. You collect from that population, and then you rebuild the riddle insert into that population. So then now when you're releasing, you're not changing the genetics. You're kind of building off what's there. That's a little bit more time intensive because you have to constantly like rebuild the transgenic insert. Although if you have it, you could mate it into the population. Like there's other ways that you could do it. But, so I don't want to say that like, they're still doing that. That was sort of some initial experimental results from early studies that I had looked at, and that has an easy fix. So I think riddle's really promising. I think the Wolbachia stuff's really promising. I'd like to see the PG SIT stuff work, although it makes me real nervous to release Cas9 alleles into the wild population, so I don't know how I feel about that, but okay, that's the end of Sterile Insect.